antibiotic-resistant bacteria are taking over, and it's just accelerating all of the time. So within 5, 10 years, 20 years, we will stand in a worldwide crisis where we won't be able to treat bacterial infections anymore with antibiotics. With that crisis kind of nearby, it's a motivator to try to figure out what is going on, what is going on at the nano level, what are the mechanisms that drive this, and how can we potentially mitigate it. I got quite interested in the field of geogenetics after I realized that there's a biological term called horizontal gene transfer. As a geologist, I did not know what that was, but bacteria are super interesting.
So we start with the project itself.
Bacteria can share genes through this process called horizontal gene transfer, HGT. This includes antibiotic-resistant genes, or in short, ARG. These genes can be transferred to other bacteria through various forms of contact. That's HGT in very simple terms. So contact is considered the most common pathway for gene transfer between bacteria. But what about DNA that's not in living bacteria? Normally, extracellular DNA or free-floating DNA in, let's say, water breaks down in days. That's why it was thought that this process for gene transfer was not important in the environment. However, our research shows that DNA can stick to microplastics and minerals, preserving it for up to millions of years. Despite being stuck on a surface, this DNA, including antibiotic-resistant genes, can be picked up by bacteria, giving them an evolutionary advantage. So antibiotic resistance ends up in our environment from hospital wastewaters, and also from farming and from local households. When we have leftover antibiotics, some people tend to pour it out the drain, and that creates a pressure between bacteria to develop resistance against it.
Piloting the boat, there's like it's basically a fish finder, and you can really see the depth underneath you, and that's actually very interesting. Yeah.
So I started this project after some conversations with Nicole Post, who was the one who actually showed me that the plastisphere is also an opportunity for microlife, and that life really colonizes on plastic particles and creates a whole new microenvironment. So we started talking and decided to run this pilot project together.
Did it go all right last week with the students? The weather was better.
Yeah.
If you have a surface like the plastisphere that's very active, and you have cells on that, and you also have organic debris, and you have metals and different minerals on it, that can also potentially be a surface that's potentially active in the spread of antibiotic-resistant genes. But the question is now, can we get good estimates of how much is there and figure out ways to reduce that, right? So it's not a continuing and exponentially rising problem.
So we tried, for now in this pilot project, 2 different types of plastic, and then we had a mineral surface as a control. And the control was really poor in propagating antibiotic resistance, whereas both the plastic particles were really, really efficient. Now we know that in the top 10 centimeters at the ocean floor, we have about half a gigaton of extracellular DNA preserved. So if you have an organism, a bacteria that is experiencing stress, it needs to evolve to survive, then we have a whole gene bank in the ocean floor, for instance, where bacteria can acquire trait if they need it. I would really like for us to understand the processes driving the propagation. Can we induce a surface that will prevent DNA from being uptaken into bacteria, or do we show that, well, this is an unstoppable process? What we really need to do is stop putting antibiotics in our system.